Hey, good morning. So we'd like to get started here. I'm sure we're going to have people coming in, but I really appreciate you guys joining us today for this is our third annual Plant Science Symposium, and we're going to hear more about what the symposium is about in a bit. But just to touch on the theme and what we hope to kind of accomplish today, the question, our theme is arms race against nature, and that we wanted to answer the question or explore the question, how do we adapt to adaptation, and particularly in the mindset of research, and that's both in the short term, how are we answering questions, how are we answering problems or addressing problems that are arising because of the changing environment and a growing population, but also how is research adapting itself to what the future needs are going to be. So we really hope our speakers today can address and help us kind of start that conversation. And of course, so with research, we wanted to get in to have our opening remarks, our Dean of the Office of Research, Dr. Kidwell. So I won't take up any more of your time, and I'm going to let Dr. Kidwell give our opening remarks. Thank you.
So why we get fired up here, first and foremost, welcome to those of you who have never been on our campus before. How many people in the room is that? Yay, welcome, welcome. We're really thrilled that you're here, and I mean that. You know, anytime I have an opportunity to hang out with people that like plants, it's a good day for me. And I got an interesting charge to link leadership and plant breeding. That was the charge. You will let me know at the end of this if I manage to get remotely close to doing that in this talk. I actually really wanted to direct my talk towards students. You know, I don't know how many of you are really crystal clear about what pathway you're going to take from here. I have to say, for me, that was kind of a challenging decision. You know, I always thought that I would be a lawyer. I'm now a dean. I don't know if there's any parallel there, but at some point along my journey, I got pretty excited about plant breeding and ended up taking a university job. But I want to talk a little bit about some of the lessons that I've learned and how that's created a platform for me to lead this college forward. So we're in the College of Agricultural, Consumer, and Environmental Sciences. I've been the dean since November of 2016, so it's not quite my second year anniversary. I'm an alum of this institution. So I'm now the dean of the college that I graduated from, which is nothing but strange. That's just weird. But I was out at Washington State University for a long time, and I was a wheat breeder out there. I was the spring wheat breeder out there. So I'm going to tell you a little bit of a story about that journey. I don't know if that works. We'll try this. So I'm going to give you a foundational book. If you've never read this book, anybody heard of this book, The Four Agreements? I used to use this book in a class that I taught, an interpersonal communications class, and I've probably owned 30 copies of this book. I'd take it with me on when I traveled, and I gave it away on airplanes all the time because people would ask me about it. And it's called The Four Agreements. The first 25 pages are weird. I actually skipped those and go to page 26. But there are 4 agreements in this book that are really kind of interesting guiding principles for life. And I learned a lot about these in my journey as a wheat breeder. So they're simple. 4, be impeccable with your word. That's pretty much do what you say you're going to do. Have high integrity. Don't take anything personally. That's really tough because most of us think everything is about us, even when it's not. Don't make assumptions. That's a big one too, you know, for those of you that have conversations with yourself before you engage with the person and have decided how the outcome's going to be, might know what it means to make assumptions. And the 4th is always do your best. In some ways, these are common sense. But what I liked about this book and what undergrads that I used to work with in the interpersonal communication arena really liked about it was memorable. Like you could remember these and try to walk them into your life. So in the beginning, so I had to make the decision, industry, academia. It was a tough decision for me because I always thought I'd work for Monsanto, you know, when I was going through school. I had no desire to go into academia. In fact, because I thought it was so dysfunctional, I just didn't want to go there. Odd that I'm now a dean, perhaps with the goal of addressing the dysfunction, which I would say is my mission in life. We'll see how long I last, you know. So I decided to go into academia because I really love to teach, and I feel like people that love to teach should do it because enough people that don't do, and it makes a difference, you know, how you show up with students in the classroom. So I decided, well, I'll see if I can get a plant breeding job in the public sector. There are 4 breeding jobs open in the country when I got out of school. It was a bad time in the job market. I got 2 interviews, 1 offer, and I found myself on an airplane heading to Pullman, Washington to be a wheat breeder, a crop I had never worked with in my life. That was not the best news, you know. So in fact, when I interviewed for that job, I had to explain to them why an alfalfa geneticist thought she could breed wheat. And I honestly said, because I don't have any bad wheat breeding habits. That ended up being really true. You know, I was trained very well as a plant breeder and a plant geneticist, but I had never applied that to wheat, and so it was a good lens. I asked lots of questions about the program, and we retooled it based on really my ignorance about how wheat really worked. But when I started, I inherited a $30,000 debt, and that was in the mid-90s, and I have to tell you, that was a lot of money back in the day, and I was not smart enough to realize that as a new person, I should not have to pay that back. So I did pay it back. Almost all of the equipment was broken, obsolete, or missing. People had raided the lab, and there was hardly a thing left in it. The thing I remember the most, there was a big graduated cylinder with a dead mouse in it. That was not a good sign. I had never worked in personnel management. I'd worked with a couple undergrads, but I'd never had to be anybody's boss. And I had a technician that had worked in that program for 28 years, and he did not like me. That was not good either. And I had never managed a budget, and suddenly I was managing one at about the tune of about $500,000 a year. That was not good either. At this point, I wasn't crying yet, though, but it got worse. I had never written a successful competitive grant. I had never created or delivered a course. I really didn't know anything about wheat, and I couldn't drive a tractor. So, not exactly the formula for success. But I will say that you develop resiliency through this journey called PhD, right? I'm a breakdown leads to breakthrough kind of girl, you know, and I had learned a lot about navigating challenge, and so I applied that skill set to a new context. And I'm not easily defeated, you know, so I had to show up with some resiliency. And I'm endlessly curious, you know. So the thing I always loved about genetics is that there are all these rules, but you never really know the answer until you see it in motion. And so, you know, I just kind of leaned in and figured some stuff out. I will say this, I still don't know how to drive a tractor. I hired a great technician who did all that for me. So I'm going to talk about the outcomes before I talk about the journey, just so that you realize from that place of beginning, I was actually quite successful. So I released over 20 varieties in the 15-year period I ran that program. And when I retired from Washington State, the wheat commission told me that they did the math. My wheat varieties generated over $1.4 billion in farm gate value for Washington wheat producers. I was stunned because I never really did the math, you know. But I got in the business of being a public breeder in wheat in particular because it wasn't saturated by industry. And the mark we made on the world with those varieties, especially in the Pacific Northwest, was quite profound. And I'm proud to say that one of my wheat varieties is still the #1 spring wheat in that region. And I've been gone for a while. And I've been out of this business since 2010. The other thing that happened was we non-exclusively licensed the highest royalty generating discovery at WSU to date. The sad thing about that is it happened in 2005. To me, that means that WSU is not very good at generating, you know, royalty stimulating discoveries. There's some room for improvement there, but it was something that I had no experience doing either, and it was kind of a serendipitous discovery. So I learned a lot about patent law and procedure, which I don't know if it's good that I know that or not. It was really kind of an interesting journey. So I did well, and there were rough spots along the way. So this is a picture of me 1,000 years ago when I was a new wheat breeder. I'd been around there for a few years. And I want to talk a little bit about how the interpersonal piece and the value piece collides with science. So I developed a wheat variety named Zach. I named it after the former wheat breeder whose name was Konzak. I liked 3 or 4 letter names. Zach was great, and I thought, wow, this is going to be really awesome because that variety in particular had incredible milling and baking quality, like nothing we'd ever seen in the Pacific Northwest. And the thing about spring wheat, it was almost like, it was kind of it wasn't a primary crop, it was like a rotational crop. And so it was the ugly stepchild to winter wheat, just because winter wheat was higher yielding. And so my goal was to create some niche markets for spring wheat because it was a great rotational crop and it was really added value in the production system, especially when you're thinking about sustainability. So we did a lot of quality testing with our lines. And this one in particular, Nabisco, we got really excited about. And the short story is it just took less water to make a really good flour out of Zach. It was much more water efficient. And so Nabisco could cut 5 feet off of their gas-powered bake line and bake really good products with it. 5 feet off of a oven baking line for a company is hundreds of thousands of dollars in savings a year. So they were excited about it, and they wanted to contract grow it in Washington and ship it down to their facility in Portland to make Nabisco products. So farmers were thrilled. Like, it was so exciting. You know, they'd never seen anything about it like this. We were getting press all over the region. You know, I was like, wow, man, we're in the big time now. I don't know if I'll ever do anything as great as release a variety like Zach in my career. It was just a phenomenal time to be there. And the demand for seed was off the top. We just couldn't blow it up fast enough for the demand for the seed. So don't make assumptions. Here's the first one. I assumed that this variety was going to be bulletproof, and it was going to be a phenomenal additional to the arsenal in the Pacific Northwest. And we had done a tremendous amount of testing on this line for resistance to stripe rust, and it had come up resistant in all the trials that we'd done. So stripe rust is the nemesis of wheat in the Pacific Northwest. It's really a huge problem. In its first year of commercial production, Zach actually showed signs of this disease. And the way that the story went, it was a Sunday morning, I was home, I got a phone call from a farmer. Farmers called me all the time, by the way. That was in the day when you had a house phone, you know, so they could just look me up and call me. Some guy called and he said, hey, there's something orange on my Zach. And I'm like, that is not possible. And he said, well, there's something going on. I don't know what it is. So I hopped in the car and I went out to my field plots, and sure enough, Zach had stripe rust. And I was like, oh, this is not good news. Not good news. And actually what happened was there was a race shift in the region. And it had blown up from Mexico. So we had seen all kinds of interesting stripe rust reactions on varieties that had been resistant before. Zach was highly susceptible to it. And what we found out from this real-life ugly earth school experience, as I like to call it, was there was single gene resistance in Zach. And it was overcome by this one race. Be impeccable with your word. So here's what happened. In 36 hours, within 36 hours of realizing we had a problem, I worked with the Washington Wheat Commission, a bunch of plant pathologists, the Extension Network, to release alerts to let people know that they had to walk their Zach and spray it. Now, this was a variety that people thought was bulletproof because it had an excellent disease resistance, insect resistance package, high yielding, great quality. So farmers were not prepared for this. Every 7 days after that, until the crisis was over, we released spray alerts to let people know what was going on. And that, to me, that was a lot about just being a good steward of the crisis. Because in a crisis, what happens is people start to disappear and the bullets start to fly, you know, and you start making excuses and trying to blame people. We chose to stand in and tell people there was a problem so they could get a plane up in the air to spray that Zach before it went to an economically devastating place, because it was burning the plants up almost from tip to ground. The other thing that happened, don't take it personally, I got 200 phone calls in 2 days. It was unbelievable. You know, my voicemail was just jam-packed. And I have to say that 95% of the calls that I got were supportive. 5% were not. The people wanted me fired or killed, whatever came first, whatever was easier, you know. And I had conversations with people that were screaming at me on the phone. And I'd have to say, I know you're mad. I need you to hang up the phone, get an airplane up in the air to spray this, and then you can call me back and yell at me later. You know, it went, and you know, they called the president of the university, they called the dean. I mean, everybody. It was insane. But I actually returned every single phone call because I thought it was the right thing to do. And I called this one guy, you know, back in the Pacific Northwest, it stays daylight till 10:30 at night in the summer. So I called a farmer at about 10 o'clock at night, and he picks up the phone and he says, "I'm so shocked that you called me back. I thought I'd never hear from you," which kind of made me feel bad, you know, that somebody would call and think I wouldn't return the call. And he said, "I just wanted you to know that this has happened to every wheat breeder that Washington State University has ever had. Everybody's lost a variety. And frankly, every breeder that I know has lost a variety. People just don't tend to talk about it. You know, those are the stories you don't tell. We tell the stories of our success, not the stories of our failures." And in fact, there was a groundswell of growers from the region that came to my rescue and actually kept me from resigning because I really felt bad about it and I thought I did something terribly, terribly wrong. Do your best. So, Zach is on the left, and you can see it's devastated by striped rust. You know, and even the theme of your conference here is about, you know, looking at situations like this because I fear things like this will happen, kind of like the T cytoplasm disaster in corn. You know, how vulnerable are we? And when I think about food security issues and vulnerability, a lot of my concern has to do with what happens to, you know, some of these plants genetically. You know, what are we vulnerable to? Because we're up against nature, you know, and sometimes nature wins. So we did our best with Zach in the moment. But what we learned from that is we had a terrible selection process for disease resistance. And so from that, we developed a whole new way to identify the difference between durable resistance and seedling resistance in these plants. We published a lot of papers and did a lot of good work that actually helped the industry adjust to this. That work led to the discovery that we patented, which is interesting, you know. So there's always the challenge point of failure versus success and what lessons you learn from the struggles. This was the most public failure I probably could have made. And how I recovered from that and dealt with that really dictated what happened next in my career. In the very same nursery next to Zach, at a much earlier stage of development, was a variety called Louise. Louise I named after my niece. Perhaps better karma there, I'm not sure. But Louise is still the #1 spring wheat in the Pacific Northwest, and that disease resistance is held up because it has both adult plant resistance and a couple of seedling resistance genes in it. Every variety we've released since then has this package, and we haven't lost one in that breeding program since. So there were some big lessons to be learned. To me, it's truly only tragic if I don't learn anything from a bad experience. And you know, you can make some big choices. I could have left WSU and forever had the shame of ending on a variety that was a failure. I chose to buck up and, you know, change that breeding program so the tragedy didn't happen again. How I navigate the situation is just as important as the outcome. That's absolutely true. And I am humble to nature. You know, nature rules, and you just never know what nature is going to throw at you. So as good as we get at selection, I'm very, very attuned to what happens in the climatic factors around breeding. And to be honest, you know, we're always students. You know, I'm often in front of people giving lectures, but I'm always a student of what I do, which I really love. You learn more from every experience that you have. So how does this impact me as a leader? So this was a defining moment for me personally, and it really did shift my perspective on how I engage with situations. So I would say as the Dean of ACEs, being impeccable with my word is one of my foundational ground rules, like having integrity and doing what I say I'm going to do. I still work on not taking things personally. You know, a lot of it isn't about me. People may not like the decisions that I make, but a lot of it's not about me. And it really is about moving the college and the university forward and our science forward in the best way that we can. I don't make assumptions. I ask lots of questions, you know. It's really important to fact check what you think is true. And I learn a lot from people by trying to figure out why they're resistant, why they're unhappy, what they want to do. It actually expands my ability to lead well. And every day, regardless of the outcome, I bring it. I'm all in all the time, you know, and I think that's important for me because it really fuels my commitment to what I do. And I'm actually eternally optimistic too, which is kind of scary. So when you think about where we're going in the College of ACEs, I got a couple slides just that frame some things, and I can't go into a lot of detail here, but I wanted to just kind of create a link between where we are and even the topic of what you guys are talking about. So we do have an overarching goal that brings the 7 departments that are in this college together, and we range from crop sciences to human development and family studies. So we got a broad breadth of things that we do. Our goal is to become the world leader in food and agricultural systems that promote global health. And when we talk about global health, we mean personal health, community health, environmental health, and economic health. We want to be the go-to place where people from all over the world come to source our expertise and to work with us to solve some of the biggest challenges that face the world. So how do we do that? Well, the first thing we're working on is expanding and synergizing the talent base. You know, we have some amazing people around here that do phenomenal work. They don't all know each other. And so part of my goal is to create opportunities for people to align and figure out who does what so we can do more together. So we're looking at the ag story a little differently than perhaps we did a couple years ago. Ag is important, it's foundational to us, but I talk a lot more about food now than straight agriculture. Looking at food as a prescription for health and well-being. Looking more from, instead of field to fork, I talk a lot about seed to soul. Like the whole system in the food production and food consumption that relates, you know, the environmental aspects, all the agronomic aspects to producing high quality food that has an impact on human health and well-being. When you do it that way, you get a lot of attention from people in that 80% of the population that just simply don't have any affiliation with agriculture whatsoever. And those are the people that we really need to get their attention so they care more about what we do. I think we spent a lot of time trying to force people to understand agriculture. My strategy is to get people to understand food and then widen that lens so they see how it actually links to agriculture and have more respect for how we grow the food that we grow. We're hiring 7 new positions, 1 in each department, that fills an essential gap around that theme of food and ag systems for global health. And that's been very exciting because that's been an all-college view of a platform that we want to stand on and bring some workforce together. It also gives us a really great excuse to align people that do that work anyway with some new energy. So we're doing this a little differently than you see people typically do cluster hires. It's going to be facilitated by Herman Bolero out of our Office of Research. So we're actually going to coordinate, invest in, and catalyze some opportunities for people that want to do that kind of work to elevate the visibility in the platform so more people know what we're doing. We also fully intend to lead that initiative out of this campus and this university, and so we're excited about where that's going to go. We're also looking at accessing new opportunities, and so instead of doing what we always do, we're opening doors to different types of things. And so we've got a couple new majors. One that actually went live this semester is a computer science plus crop science major. So it's kind of that data analytics blend of bringing science and data together. So that's kind of a new interesting twist. You know, data analytics is a big deal. This is the first major of its kind in the country, and we hope we gain good traction with that. We're also developing a metropolitan food and environmental systems major to target people from urban areas that are interested in food and backing that one into agriculture, so that's in a build phase. We're in the process of developing a concept note to build a food innovation center in Chicago. One thing that we need to do on this campus and this college more than we have done in the past is create more visibility in urban populations, and so we're really targeting the opportunity to expand and gather more interest and engage in more opportunities, especially related to extension in some urban areas. And we're also going to hire an Associate Dean of International Programs to elevate the value and importance of that particular portfolio to the college, to the institution, and the contributions we can make in global health. The last thing I'll talk about is elevating research and extension efforts. And so this has been a problem all over the country, and I think, you know, for students we see this a lot. You know, there's a lot of great basic research that we do, and sometimes it's very hard to figure out how that does anything that makes a difference in the world. You know, it's a big leap, you know, and we all say ... solve world hunger. If all the ... solve world hunger that all of us have said for the many decades it's been said were true, we wouldn't have a problem, right? We still do. So we want to make sure that the discovery work that we do has some translatable opportunity to create transformation in communities, in industry, in science. So we're really focusing on doing relevant work in a way that it makes a difference. So to do that, we've got to close that gap between discovery and application. It's an interesting time to do that. You know, I think the public outreach, the extension work is so important, and it's lost a lot of funding over the last few years. So we're investing in applied research, extension, and outreach because we want to be relevant to the public, and we want the public to know that what we do matters to them. Part of it is telling our story better. We also have to figure out how to acknowledge people for contributing in those areas. And so the last thing that we're really focusing on is connecting with the public 2 ways. The public coming to us for support and engagement, and us going to the public to create more of a closed loop system around how we feed and fuel each other. So for me, so many of the lessons that I learned in plant breeding have really shaped the way that I'm trying to step into a leadership role in this college. And I say this sincerely, like I stepped into this to make a difference because I didn't necessarily like what I saw. And I encourage you to think about that too. One way to be the change you want to see in the world is to buck up and do it yourself. And to do that, sometimes you have to step into a situation, play by the rules that are established, be successful in that system, and then change the game. So I feel like for me, that's been the platform that I've transcended from. But my time as a plant breeder was absolutely sacred to me. It was a fabulous, fabulous career. It was the hardest decision I've ever made personally to let go of that and become an administrator. And now I have an impact in a different way. But those are cherished, cherished memories. And you know, my former buddies from Washington still call me, and I consider that to be the true test that I made a difference there. So they're in the combine telling me how good things are yielding or yelling at me for what's not going well, still, all these years later. So with that, I'll answer a couple questions if you have them. If not, I really hope you have a fabulous, fabulous conference, and we truly are thrilled that you're here. Thanks for coming. Questions? Questions, comments, thoughts? Yes, sir?
What was that plant on the cover of the book that you showed?
You know, I don't even know. To be honest, I haven't looked at it that closely.
I mean, corn country wasn't corn.
Yeah, I don't know. I don't know. That's a good question.
It's like a dandelion.
Yeah. Yes, sir?
What's the story on the author of that one?
Well, you know, he's kind of a, I would say he's a life skill guru in lots of ways. He writes lots of books around self-management, self-awareness, and so this book came from an interpersonal communications course that I taught. I was the wheat breeder that taught the most popular communications course on campus. Long, long story. But I came across him in some trainings that I did. Somebody said, hey, read this book, and I really liked it. So he's written a lot of books around topics of, like The Mastery of Love is one of his books about relationships. That one was a harder read for me. This one was better.
Are you providing a free copy there?
I am not. I am not, but you know, it's an interesting thought. Yeah, interesting thought. Other questions? All right, thanks so much.
Thanks, Dr. Kidwell. I really think that set the tone for today, just about how we can, you gave us a bit about your past and now how you hear the future today. So as many, I mean, part of the future is our future grad students, including myself. And one of the ways that we're getting ready for the future is by building skills, and one of the ways we build skills is this opportunity to plan this symposium for you guys. So we have Jesse Monkford here from Corteva Agriscience. So the symposium is 100% student organized, but we are super happy that we have the support and sponsorship of a company like Corteva. So please welcome Jesse.
Thank you for the introduction, and Dean Kidwell for that nice intro talk. I think Brian's right that it really sets the stage for today. So my name is Jesse Monkfold. I'm a senior research scientist at Corteva Agriscience. And I'll have to say this symposium series that the university is a part of today is really possible. That sponsorship is possible from a lot of work of Jason Rauscher and Tabare Abadi. And Jason's here in the audience today, so you can get to know him. They work extensively with universities and other institutions to support and help the organizing committees put on this symposium series. So I don't want to spend a lot of time today. I know you're eager for the other talks, but I do want to set the stage a little bit. And there are a couple extra slides that we need to show. And that's because we have a different name. And some people may not know the history of where that name is coming from. About 1 year ago, just a little over a year ago, Dow and DuPont, the larger companies, merged and finalized that merger. And the intention is to bring together those two companies and to spin out 3 separate companies. one of which will be an exclusively agriculture-focused company. And that brings together Dow AgroSciences, DuPont Pioneer, and DuPont Crop Protection. That company, which is intended to spin out sometime middle of next year, is what is named Corteva Agriscience. And so it's a new name because it's a new company that brings together a lot of capabilities in different specialties, but we're also going to have a revised purpose and vision as Corteva. And that's what I wanted to touch on here. There was a lot of thought in bringing these companies together about what is our future purpose, what is the culture of our company, what are we aiming to do? And that comes out in this purpose statement here: to enrich the lives of those who produce and those who consume, ensuring progress for generations to come. And I've had the opportunity to think a lot about this purpose statement, and it really has begun resonating with me. And the reason is for a couple of the things that are in here. To enrich the lives goes beyond just a business purpose. It goes to this bigger purpose of improving agriculture, improving the lives of people around the world. And it also includes something else in here. You notice it's those who produce, those growers, farmers working the field, but also those who consume. So a larger picture of partnering with society, not just the people that use the products, but the ultimate consumers who are the ones that are being fed by the food that that farmer is growing. And so I think Dean Kidwell's talk, and especially the end part, connects very well with this, talking about our purpose as Corteva. And something you may not be aware of is that Corteva and the university have a strategic relationship that's been ongoing for a number of years. And the strategic relationship is looking at how can we collaborate together in 4 different areas: in research, in education, in economic development, and in outreach. And so we're looking continuously for ways that us as a company and the university can go further in those areas. So Corteva, this is just a little bit about the company by the numbers. We'll be a very balanced company when we look at the 2 main technologies in seeds and crop protection, almost 50-50, over a wide range of crops and products. 22,000 people strong, a global organization in 130 plus countries. And that matches this symposium series. You know, this is really remarkable, and Dr. Kidwell and I were talking a little bit about the scope of this program, that since 2008 there have been more than 128 events hosted by 50 institutions representing 13 countries on 6 continents. That's a lot. Those are a lot of symposiums and a lot of people that have been able to engage in this forum. So the objective of this series is really to empower future scientists through supporting and enhancement of the graduate education and networking, build student leadership, and to expose students to a wide range of career opportunities and topics in agriculture and the plant sciences. So how does it work? Corteva sponsors and supports, but not exclusively, and it's fully student organized and run. So everything that happens today, you know, we support in any way that we can, but it's really the students and the great organizing committee that have made this possible. So just a little more, students have full control. They decide what the format and the purpose of the event that they're organizing is, how they're going to proceed with sponsorship and budgeting, timing, communication, web pages, all of that is organized by students. So what are the topics in these symposia? Well, breeding, as is one of the topics today, is of course a major topic, but you can see some of the other things that have been touched on over the years. Genomics, phenotyping are big, new technologies such as CRISPR, and then also high throughput in all of those things, cover crops, a wide range of topics. And again, these are student selected and organized about what they feel is important for them to address in the symposia that they organize. So this is a network. So we have not only this symposia, lots of others around the world. We have speakers that go from one institution to another to participate in their symposia. And really the value in that is that it creates this network around the world of people that participate in these, that get connected, and that can further use that network that they've developed as they move on in their career. So where do the speakers come from? Mostly from universities. That's where most of the speakers are. We have some that come from industry. These are all the organizing committee chosen, and so they find speakers that represent the topics that they want to cover. But primarily the symposia really address the work that's going on in the public sector. And so you can go and find more information about the other events that are happening. There's right now, it's still a DuPont Pioneer webpage. We're working through a lot of that integration component, so at some point I'm sure that will change over to a Corteva AgriScience webpage. But then also the Facebook page has information on the symposia series in general. And today is a special day. Right now, we have 4 symposia around the world that are going on today. We have the one here in Illinois, we have one at Simmet, one at ICRISAT, and one at Kansas State. And so we're looking at today as possibly being the record for number of attendees on a single day for this symposia series. So we'll be taking count here as well as all the other sites so we can see how many people were able to connect to this today. So with that, just a brief introduction. I think we're all excited to get to the wonderful lineup of speakers that we have today. If you have any questions, Jason's email is right there. Any questions or suggestions, he'd be happy to address those. And just thank you everybody for coming, and special thanks to the organizing committee for putting together such a wonderful event.
So it is my pleasure to introduce today's first speaker, Dr. Sarah Hind. Dr. Sarah Hind received her PhD from the University of South Carolina, Columbia, where she also did a postdoc. Later, she did a postdoc at the Boyce Thompson Institute, where she also became a research associate there. She joined the Department of Crop Sciences as an assistant professor in 2017, where her lab studies how the plant immune system detects pathogenic bacteria that cause diseases in tomato and vegetable crops. So please join me in welcoming Dr. Sarah Hind.
I'll just use this mic. Sure works. Excellent, thank you. I want to first just thank the organizers. They did a wonderful job, and I was really excited that they asked me to come and give a talk. I also warned them up front. I said, you realize I've been here a year, and we don't have a whole lot of data yet to show. And they said, oh, that's fine, you can set the stage. So that's really what I'm going to try to do. We've had 2 great speakers, Dean Kidwell and Jesse Munkfold, who have set the stage about the symposia, about research, and now I'm going to dive into the science. And so I really took to heart, what does this mean by this arms race against nature? And so I'm going to talk a little bit about pathogens, because that's what I'm working on, but then I'm going to talk a little bit about genome editing, because I think that's going to come up a lot today, and then where I see my program going, what I'm going to be working on. So I have a lot of parts, and first I'm just going to give a background, because I don't know if everyone here has pathology by training. I'm going to guess not, so I want to make sure we're all on the same page, talking both about disease and then also about the plant immune system. And then I'll go into genome modifications and kind of give you an update on where we are. I tried to pull from the most recent papers to give you an idea about how fast we're moving when it comes to development of technology for genome editing. And then finally, I'm going to be talking about Xanthomonas. I'll give you some background about this bacterial pathogen that's has a lot of economic importance. It infects a lot of our crop plants. And then I'm going to talk specifically about the program that I'm starting to set up here at the university, focusing on Xanthomonas, which causes bacterial spot of cucurbits. So first, plant disease and immunity. So hopefully at some point you've seen this concept of a disease triangle, where in order to actually cause disease, you have to have the right conditions. First, a favorable environment for the pathogen to be able to infect the host, to be able to replicate, to be able to disseminate. So there's some factors here that unfortunately we can't control, such as rainfall or humidity, but then there's other factors such as soil fertility, potentially soil pH, that we're able to hopefully manipulate and make it less favorable for some disease development. On the pathogen side, when we think about microorganisms, the vast majority of microorganisms are not pathogenic. They are not able to cause disease. It actually takes just having a certain genotype, the certain ability to infect. It's rare that actually microbes can cause disease. So you have to have just the right pathogen on the right host. And then talking about the host, again, most host plants are going to not develop disease symptoms when they encounter these microbes. It really takes a host that is susceptible, and this can be at the family level, a whole family of plants. It can be an individual species. It can be an individual cultivar. So it really depends on a lot of factors in order to actually get disease. And so thinking big picture wise on the factors that actually influence our crop production. And so I'm sure we're going to hear more about this, but two that we can focus on. The first is this idea of, if we want to try to help control pathogens, really the best way is to do polyculture. And I know that doesn't happen here in the Midwest or in a lot of places where we do large production, but the idea is if you have mixed plants around, it's going to be harder to build up large inoculum and be able to spread it. So that's one way, in theory, you can control disease progression. The other is crop rotation, which is what we do since we have a lot of monoculture crops, where we rotate through using different plant families so that we don't have a lot of pathogens overlapping. And this gives you a time for the pathogen load to leave the soil or the environment. Biological control, I'm sure we'll hear a lot today. Biological control, chemical control, the idea that we're able to treat our crop plants with chemicals to prevent, say, weeds or bacterial or fungi, but then there's also biological controls, and this can be anything from spraying plants with a beneficial bacteria to try to prevent disease development, or if we're thinking about insects, then this can be introduction of predators to be able to eat those insects that are feeding on your crops. And finally, controlling movement. And we often don't think about this, but it really is important for these phytosanitary precautions that we don't move these pathogens all around the world. Unfortunately, it still happens. So I found this great paper where it was looking at first reports, meaning first documentation of a pathogen in a new location. And so this is just 6 years. But all around the world, you see that, again, with a lot of global transport, we're bringing these pathogens all around the world. We're introducing them to new places where they can find a favorable environment, they can find susceptible cultivars, and then these diseases can spread like wildfire. So this is an area, we're trying to do a good job, but obviously there's a lot of room for improvement. It's also pointing to the fact that we have really good surveillance mechanisms in place, right? So we're able to keep an eye out and look for these new introduced diseases and then try to catch them early before they really get a foothold. And I'm going to talk about an example later on of a Xanthomonas that got a foothold, and it's a Xanthomonas pathogen of banana, and it's just wiping out banana production in Africa. So we'll talk about that one later. So I kind of covered some of the others. So by focusing in on crop protection, by focusing on pesticide, herbicide use, management of our crops, that allows us to minimize the potential loss that we have. So in this paper, it just really gives you, first of all, this scenario: if you don't have any crop protection, you're not using pesticides or herbicides, obviously you have a significant potential for loss due to not only disease-causing agents, but weeds. We won't forget weeds. I know we have 2 weed science talks later on today. We're not forgetting the weeds. They're very important. So of course, when we utilize these management strategies, these crop protection strategies, these pesticides, we're able to boost our production, minimize our losses. The only other thing I want to point out, actually, it is really, if you look at the weeds, it is really significant how the herbicides really are saving us from having a huge amount of crop loss due to weed problems right now, even more so than the pathogens and the animal pests. But I'm going to focus on pathogens today. So it's still a significant problem, and if we can better manage disease, then that's going to really boost the amount of production we can have. Great. So just bringing this back. So again, I kind of touched on some of these favorable environment, a little bit about pathogen and host, but now, of course, now I'm going to dive more into these pathogen and hosts and how they interact. So there's a very dynamic, complex interaction between a pathogen and a host. I'm going to focus in on bacteria because that's what I'm going to talk about today. And a lot of bacteria are able to not directly penetrate into the cells, but often they're able to transmit proteins or other molecules that are able to manipulate their host. And the system that I'm going to talk about today is this type 3 secretion system that many pathogenic bacteria have. Think about it as a molecular syringe. Since the bacteria can't get into the cell itself, it lives in the apoplastic space. It uses this syringe-like molecule to cross the plasma membrane, and then it's able to move proteins from inside the bacterial cell directly into the cytosol of the host cell. It can transmit these proteins. We call these bacterial effectors. These wreak havoc in the plant cell. And so initially the plant can respond. It has an immune response that we call pattern-triggered immunity, or PTI. So this is the plant is able to recognize some signatures of the pathogen so that it knows it's under attack. These effector proteins are really good at targeting aspects of the system and shutting it down. And so that's what we're seeing here is that, the pointer's not great, but initially we have a response to the pathogen. And then this can kind of, if you have a bacteria that's not able to cause disease, what would happen is that the plant would recognize it and it would shut down that bacteria before it had a chance to cause disease. But for those that are better adapted, those pathogens that are better adapted, they're able to overcome that with these effector proteins and suppress that immune response. Of course, this is an arms race. We're going to talk about a lot of arms races today. And so in this ongoing warfare between the host and the pathogen, a lot of host plants have sort of evolved additional surveillance mechanisms to be able to detect these effector proteins that are coming from the host. And this is what we call, resistance genes. And so this is a second layer of the immune system. It's much more robust. And this is where we get that programmed cell death or hypersensitive response that you hear about. And so this is extreme because it's actually cell death, but it's a way for the plant to robustly shut down that pathogen. Sometimes that's kind of the last resort. So that's our general system. When you start looking at these effectors, it's fascinating to actually study the effectors. Is that what I pushed? What did I push? It's fine. I'll keep talking. So it's fascinating to study what those effectors target because they tell you what's important about the host plant, right? So a lot of scientists have studied effectors and followed what they target and use that to tell them what's important about the plant. Did I push something or did it just? Okay. I didn't think, wait. Something's there. So again, we're going to come back to this. It tells us, they can tell us what's important. And there's some sort of obvious candidates. Again, they'll target the immune system, the receptors of the immune system, some of the signaling molecules. Other things that don't seem as obvious, they often manipulate the hormones. And so that can cause, aberrant growth or development of the plant. They can target gene expression. So again, they can turn on or off genes that would make it more beneficial. They also target the cytoskeleton. So the cytoskeleton plays an important role in helping move toxins once they're produced in the ER, and then they're going to be secreted through the secretory pathway. Hooray, you got it. The cytoskeleton plays a role in directing those vesicles to the plasma membrane. So if you interfere with that secretory pathway, you're not able to get those toxins outside to start inhibiting growth of bacteria. And so again, you can use the pathogen to tell you what's important about the host. So this is as much as we're going to kind of go into, but I want to show you it's actually way more complicated than I'm showing. This is actually 6 years old now. I would love to see an updated version. I haven't seen it, probably because it would be really hard to do. But again, these are just focusing in on bacterial type 3 effectors. There's a whole other suite even of bacterial proteins that are important, but these are just the type 3 effectors. And again, they're targeting every aspect of the plant cell to try to shut down that immune system. And it's really fascinating to be able to, again, use this to tell us what's important. And I want to see a review. I don't know who we need to talk to get to do a review, but it would be great to see what progress we've made in the last 6 years in our understanding as well. Okay, now I'm going to jump a little bit over to breeding. And I think we're going to talk a lot more about this, but I just wanted to bring up this idea of, traditionally, if we were trying to bring in multiple traits, we would do some sort of gene pyramiding scheme, right? So the details are going to be different, but the idea is that if we have different parents with different traits, we're going to try to breed, cross them, breed them together to try, in the end, to bring all those traits together. Looks really nice on paper, and I'm not a breeder. We can probably ask Jesse, and he would tell you that this is way more complex and difficult than we can make it look on paper, right? When we start trying to add more and more traits, it gets really difficult to do, to still maintain all the other traits, maybe yield or drought resistance, other factors, trying to maintain sort of that elite background while trying to bring in new traits. It's very, very difficult to do. There's a couple examples that I wanted to just pull out. So you can do this for disease resistance. These are cases, again, where they did traditional breeding to try to pyramid in some, on the left it's more resistance genes, so you can see that you have significant reduction in symptoms caused by this disease. On the right, what we're looking at is it's more QTLs. So again, they're trying to bring in 4 QTLs from this parental line into their background. So again, you can do it, but the more genes you have, the more complex it gets. And so what we're looking at now is there easier, better, more efficient ways of doing that than trying to go through, there are examples of this working, but is there more efficient ways of doing this? Especially, again, as we have these elite breeding lines, we don't want to mess up their background too much, right? We just want to try to add traits without messing up things that we already have gained. So one way that we can start doing this is the idea of actually using genome editing, genome manipulation, and gene stacking, where we can bring in multiple genes at once in one area of the genome through direct targeting and through direct manipulation. So this is an example I made. They could be any genes, right? You could bring in a whole slew of them, and they've actually started to set up systems where you can integrate some genes, but they have a specific site in them. So later on, if you want to add another gene, you can just pop it into that site. And so that's actually what they're showing here. Let's say you have a plant that has these 3 genes, say 3 resistance genes. You want to add a new one you just discovered. You can clone it on a plasmid, and it's able to actually integrate over here because you have a specific site where it's able to recognize and pop it in, right? So something like this would, again, it would be great for these breeding lines where we could introduce genes and then add new ones when we want to without having to do extensive breeding and back crossing. So that brings me to genome. How do we do this? How do we actually do these genome modifications? I'm sure everyone is pretty familiar with transformation, agrobacterium-mediated transformation. Of course, the problem is that when we do that, we have a lot of the bacterial signatures there, so those are going to be considered genetically modified, or GMO, under the USDA. So I want to talk about some of the strategies we might be able to do going forward where we can eliminate some of those foreign DNA signatures while still being able to bring in potential new genes that could improve our crop plants. So here's kind of the big 4 ways that you can do genome editing, and they kind of go from oldest to newest as far as left to right, and also the ease and feasibility of integrating them. So we've learned a lot over the last, say, 10 years, 15 years about how to do it, and we found better and better ways to do it. And so it started out with these meganucleases, which are engineered restriction enzymes, quite large. Then it moved to sort of zinc finger nucleases, which in this case, each nuclease recognizes a specific 3-base codon, so you have to have a series of these nucleases to have a specific DNA sequencer targeting. And then we moved to talons, and I'll come back to talons because these are actually from Xanthomonas. But talons, they manipulated so that each repeat is able to, again, recognize a specific nucleotide. And then they added an enzyme at the end to do a cut in the DNA. And then finally, CRISPR. We'll talk a little bit more about CRISPR. So the first 3 are large proteins, and you have a protein to DNA interaction. And again, it causes these very large proteins to be able to do this. Whereas CRISPR, you have an RNA to DNA interaction. And so while CRISPR is big, the actual recognition sequence is quite small. But in all cases, these cause a double-stranded break. And this can lead to 2 outcomes. The first is the most common, and that's the plant is scrambling and just trying to fix its DNA, right? And so this is where you get insertions, deletions. They can be small, they can be larger. But it's just the plant trying to stitch those 2 pieces of DNA back together. Again, this is what the first sort of generation of CRISPR mutants, that's because it was cutting the DNA and then you got these random incorrect corrections of the DNA. Where we're really trying to go now is this idea of having homology-directed repair. Now this can happen from the other chromosome, right? The other chromosome could be used to correctly fix the broken DNA. Or you could introduce a template that has regions of homology on either side of where you're going to get the breakpoint. And then that break will be repaired using that new template. And so this is how we're going to be able to insert new sequences, different sequences, potentially in whole genes. Okay, so just the real basics. I didn't want to get into too much detail with everyone because it gets really complicated fast. But basically, this is just showing the CRISPR enzyme. And again, it has this short recognition stretch, and it has to be really precise. So this is why DNA is known as high specificity. You can't have a lot of mismatches, otherwise it doesn't recognize it. And so you'll get this double-stranded break. It'll cut on both strands. And that's how you are able to then get these incorrect repairs. And again, just mentioning that the homology-driven repair, where you have a template to fix it, is very unlikely to happen. It's usually maybe 1 or 2% maybe of the repairs. Most of the time it's this non-homologous end joining which just stitches it back together. Okay, so this was CRISPR as we understood it for many years. But of course, we're never satisfied, right? Scientists are never satisfied with the same old, same old. So over the last several years, there have been scientists that have thought of new ways. How can we use CRISPR in new and innovative ways? And this is kind of the state of the toolbox of how we can use CRISPR. So again, the one I went through on the top left, the gene editing, is just what I said, right? We're using this RNA sequence to target and to cut double-stranded break. They've also done it so that they have made the actual Cas9 enzyme inactive, and they hook it up instead to other enzymes that can do base pair editing. So this is a way to change a SNP in a very specific region. And so again, you can hook it up to different ones that either go, A to T or C to G, but it allows you to, within the recognition sequence, to change a single base. And sometimes it's all you need, right? Sometimes it's all you need to be able to have an outcome that you're looking for. And so this is a very precise way, again, using that specificity, it's a very precise way of editing single bases in the genome. There's also this new one, it actually came out in the last year or two, where there's a different version. Instead of Cas9, it's Cas13, and it actually edits RNA. So it actually edit your mRNA. Again, it's single base changes right now, but of course, they're gonna modify that. But right now, it actually targets RNA. So you can actually keep your genome intact, but affect the sequence of the mRNA. Gene regulation. So again, they're taking an inactive Cas9 enzyme, they're hooking it up to either a an enhancer or a suppressor of expression, right? So you can either turn on a gene, turn off a gene. So instead of targeting something to be edited, you're targeting the protein to land, say, in the promoter. And so it'll land in the promoter, and if it has an enhancer, that means that you'll get higher expression. And if it has a suppressor, it's going to inactivate that gene expression. So this is a way of sort of subtly modifying expression without doing sort of long-term genome editing of that promoter sequence. And actually the same sort of thing can kind of happen with the epigenome, right? So what they've done here is, again, hooked up an inactive cutting enzyme, Cas9, to these epigenetic modifiers. So these can be methylation or demethylation. These can be histone acetylation, deacetylation, whatever to your heart's content you want to do, you could hook up these epigenetic modifier proteins. And again, because you have that very specific targeting, you can get it to right in this place in the genome you want to change. And this is going to be a little bit longer memory, right? For epigenetics, when you start putting on these methylation marks, it's going to be a longer term memory of changing gene expression. So there's a couple other cool ones. So chromatin topology and chromatin imaging. And so with the topology, the idea here is, and Maze people will be able to understand this, often you have these extremely distal regulatory elements. And so it's all about how the chromatin folds to bring those regulatory elements close to the promoters of the gene, right? And these can be several kb away. So what you could potentially do is use these Cas9, inactive Cas9s, to bind your regulatory element and your promoter, and then they have a dimer subunit that'll come together. And so it'll sort of artificially bring that regulatory element right by the promoter more constitutively. And then with the chromatin imaging, it's the idea of being able to tell where in the cell you have certain genes expressed, and you can follow, they're doing live cell imaging, so they're following it during mitosis, meiosis. It's really pretty cool. Again, a lot of this is being done in mammalian fields, but we'll be able to translate it over to plants over time. So this is where we're going, right? It's beyond just sort of mutations. It's allowing us to do really tailored modifications of the genome. So the part that I'm really interested in, I hope I get you excited about though, is this idea that we can insert new genes. So we can go in, we can replace maybe genes that are non-functional or cause susceptibility of the host. We can exchange them for new alleles, for new variants, introduce whole new genes. And so you can do this, obviously, whole gene. There's an example that came out in rice a couple years ago where they actually, it was quite smart, they just needed to change out for a new exon. There was a SNP in one of the exons that they wanted to change out. So they actually did 2 double-stranded breaks, one on either side of that exon. And then they had homology, in this case it was non-homologous end joining, but it popped in that new exon in place. So it kept the whole gene intact except for that one exon they wanted to change. And the other way they did it was they did a single break and then they inserted all the rest of the exons with that one exon that had a change. So there's kind of different ways of doing this. So again, it's not just maybe whole genes, you might be able to just swap out certain parts of the gene. That could be helpful, especially if you're going really, say, monocot to dicot. Sometimes you have issues moving genes that far apart. Sometimes the elements of the proteins just don't work. And so this might be ways of sort of maintaining parts of the protein that are important, but then changing out, say, the recognition domains or so forth to be able to translate it over. And how do we do this? So again, sort of the standard way is to go back to agrobacterium and use it to transform the plants with the DNA that will encode the CRISPR and the Cas9 and that template. And then you have to go, of course, through your regeneration, your T0, your T1s. And what you need to be able to do is find plants that have the genome modification, but get rid of the CRISPR-Cas9 DNA. And those are not linked, right? So your Cas9 can integrate anywhere in the genome. You're going to have your mutation in a very specific place. So then it's just a matter of numbers games where you just have to go out progeny to try to find ones that are homozygous for the mutation that don't have that Cas9 cassette. Other ways that people have been starting to go is the idea of transiently expressing that CRISPR part system. so that it never integrates into the genome, but you still get the genome modification. And so they've done this in protoplasts. You could do this in plant tissue culture and then regenerate plants from there. And you wouldn't have to go through those extra generations to get rid of that Cas9 cassette because it would have never integrated. All you had was the genome edits. So again, a lot of this still depends on being able to get transgenics. That's, I think, the one downside. But it's going to allow us to do a lot of really cool manipulations of our genomes. Whew. Okay, get over the glazed look of too much stuff there, but hopefully you get a sense that there's a lot of really cool genome modifications we can now do. So I'm going to talk about Xanthomonas, which is the pathogen that I'm working on, and trying to explain it's really important. So it's a pretty big genus of plants. So there's 29 species that are currently labeled as such. They're kind of add a new one each year as they start to reorganize the phylogeny. But right now there's 29 species. They infect about 400 different plant species, broad across the plant kingdom. And so Xanthomonas is a gamma proteobacteria. It's gram-negative, and it causes sort of a very yellow, mucilage, sticky kind of cell. So if you've heard of xanthan gum, if you ever look on the back of the package, so xanthan gum is sort of a binder that comes from Xanthomonas. Okay. What's kind of cool about Xanthomonas, there's sort of two big lifestyle types. The first one is Xanthomonas that live predominantly in the xylem, so in the vascular bundle cells. Then the other type lives in the mesophyll, so they actually live in the spaces between the mesophyll cells. And I say different lifestyles because the ones that are in the vascular bundle, they tend to clog up the xylem and cause wilting disease or rot, right? You're not getting water to go through your xylem, the plants collapse. In the mesophyll pathogens, you get more of spot disease or blight, so you're getting some death of cells or but it's not widespread kind of necrosis, sometimes killing of the plant. Now, it's not just that the, say, vascular pathogens are genetically really different than the mesophyll pathogens. Because if you, I know I apologize for the blurriness of this slide, but if you look here under the vascular pathogens, there's Xanthomonas oryzae pathovar oryzae, which causes rice bacterial blight. And then under the mesophyll, there's Xanthomonas oryzae pathovar oryzicula, which causes leaf streak. So these are genetically very similar, right? Because they're the same species, but they're able to have very different lifestyles. And when you look at the genome, there's not very many genetic differences. It's actually a gene expression difference. So genes are differentially expressed, and that's what's causing the lifestyle changes here. And so there's a lot's going into trying to understand what's controlling these different behaviors. But as you can see, they infect monocots, they infect dicots. This is kind of how we currently understand it. So some of these species are very host specific, very tissue specific, very host specific. I'll give some examples because I work on tomato as well. So there are 4 Xanthomonas species that infect tomato. There is Xanthomonas perforans and Xanthomonas uvesicatoria. There's also Xanthomonas vesicatoria and Xanthomonas gardneri. And all 4 of these species are specific to Solanaceous crops. So very host specific, and they're all only non-vascular, so they affect the leaves, these mesophyll pathogens. Switch over, you look at this Actinopodius. It infects monocots, it infects dicots. It can be vascular, it can be non-vascular, right? So we're still trying to understand how this all works, right? How is host range determined? How is specificity of the tissue type determined? But it just shows you that there's a lot of genetic variation, a lot of things we can learn from these pathogens. And then again, as you're looking, all the different families that Xanthomonas can affect, right? It's pretty much crossing the plant kingdom. And I'll talk a little bit more to focus in right here, really these tails. So I'll talk about what these are, but you can see there's yeses and nos down the board. So these often are really important for virulence, but you don't always need them. There's a lot of strains that don't have them. There's other strains that have 20 or 30 of them. So it's kind of interesting, again, how it has all these different strategies. So the example I wanted to give for this one is Xanthomonas wilta of banana. And you can see, as I mentioned, sort of that yellow mucousy color. So that's what we're seeing here in the leaves. You see it in the stems, you see it in the fruit. Quite nasty disease, very difficult to control. So this was sort of introduced into East Central Africa, really starting around 2001, and it quickly spread around what they call this lake region in Central Africa. And again, I apologize for the blurry one, I couldn't find a better one. Even in this, say, 15 years or so, actually this was in 2009, so it was only, for some places, 8 or 9 years. In red, these are areas where more than 80% of the banana farms had this wilt disease. So this spread very rapidly, and there was really no control other than getting the plants out of the ground, burning it, letting the ground be fallow, or doing some treatments of the ground before you put any new plants in. So again, banana production, it's not just the dessert bananas that we eat, but it primarily is also these cooking bananas that are providing a lot of carbohydrates for diets of subsistence farmers in this area. And so there's significant production. And if you look here, that this was the average loss of banana production due to this wilt disease. Some places like the Democratic Republic of Congo, over 80% loss. So this was a significant pathogen affecting this region. It still is. But they're making progress. And so they did that through genetic modification. And so this is an example actually of a field trial that they had. Over here on the left is a transgenic banana. And you can see it looks very nice and healthy. You don't see any evidence of this sort of gum in the vasculature or in the fruit. Whereas here are the control plants. You see they look awful. And so this was actually a case where they took 2 genes from sweet pepper that were known to be effective against xanthomonas in pepper, and they put them over in banana. And you can see the results. It was amazing. So there's actually other genes from, again, other crop plants, or I guess Arabidopsis is not really crop, but other plants that potentially could be, say, moved over to banana to be able to help control this disease. And there's ones, they've been tested sort of in greenhouse conditions, but they haven't been moved to field trials. So there's a lot of place for this to move, but it really, again, relies on transgenics, right? There's no sort of natural resistance in banana, so they're trying to bring it from other plants. So these tau effectors, so these are called transcriptional activator-like effectors. They basically mimic transcription factors. So they're bacterial proteins in xanthomonas that get injected into the plants, just like the other effectors, but they go and manipulate gene expression. And so for those genes that they manipulate that cause disease or more disease symptoms, we consider those susceptibility genes, or S. So they're the opposite of our genes, right? Now again, just like some other plants, there are some plants that have evolved resistance genes that have sequence similarity to the susceptibility genes, so that the pathogen inadvertently turns those on. So that's one way the plant is able to overcome these. Or there are plants, of course, that have, say, mutations in the recognition domains, right? So these are promoters. They're binding in the promoters. So you have changes in that promoter sequence, so that tau effector can no longer bind to that region and activate transcription. So there have been people that have started to engineer resistance, right? So you can imagine you can take some of these regions that you know that the pathogen has and engineer an R gene that has these recognitions. And you can actually stack them, right? You could stack some of these elements so that multiple effectors, potentially from different strains, could be able to recognize those and turn on the resistance gene. So it's quite complicated, but it's actually really cool. So it's these repeat domains, and there's 2 variable amino acids in these repeat domains, and those 2 amino acids encode a site that's able to bind to a different amino acid. So it's kind of a cool way of being a transcription factor, being able to bind DNA. So some of the known targets, transcription factors, that makes sense, right? That they would want to bind transcription factors, turn them on, and manipulate the host that way. There's another class of proteins called sweet genes that are pretty common targets of these. These are sugar transporters. So they get expressed, they go to the plasma membrane, and they pump sucrose out to the outside of the cell. Perfect for a pathogen that's living outside the cell. And so we're just starting to understand what these are. Again, some strains such as Xanthomonas oryzae, pathophyra has like 20 or 30, so we're trying to understand what they do. For this, again, we go back to this idea of an arms race. So initially, the bacteria has one of these tails. It can turn on the susceptibility gene, and you get disease. The plant can evolve to have potentially changes in that recognition or resistance gene, so now the plant wins. And then the bacteria again evolves, maybe the tau effector duplicates and has some changes, neofunctionalization, or it no longer can bind to the promoter of the resistance gene, right? So there's ways, so this is ongoing evolution. The question is, can we do something about that as scientists? And so what you see some scientists doing is they call it talomes, because everything's an ome, right? Proteome, metabolome, so there's talomes, where, especially common in rice, because there's a lot of rice pathogens with these. They'll sequence a whole bunch of strains, pull out all the tau effectors, and say, what do they target? And they're looking for basically what they call susceptibility hubs. So are there certain genes where lots of different tau effectors target them? Those are the genes that are going to be really important for us to manipulate, either through looking at germplasm for changes in those recognition domains so that we could maybe breed that in, or through genome editing, where we can sort of change those sequences so that those effectors no longer bind. So this is kind of the direction that they're going, again, trying to figure out what are the most important susceptibility genes for disease and changing them. Okay. The last part. So where we are with bacterial spot disease and kind of what we're doing right now. So pumpkin. We're a pumpkin state, right? There's a lot of genetic diversity in the cucurbit species. There's 11 species. There are 5 different domesticated species, though we really commonly grow 3 of them. And they had different origins of domestication throughout the Americas. I'll go kind of quickly through this. I just love the pictures. So Cucurbita maxima is one that we commonly grow. And then we have 2 wild relatives that came from either the Andes or Ecuador that are quite small. The fruit very seedy, so you wouldn't want to eat them really. But that's kind of probably the progenitors. Down, oops, sorry. Kind of down again here in the Andes region or more on the coast of Ecuador. We also have a moschata, and I'll bring that one up. So Cucurbita moschata is the species that our canning pumpkin belongs to, so it's quite important for what we're doing in my lab. And the really common one is Cucurbita pepo. So that's jack-o'-lanterns, summer squash, zucchini. So it's a lot of things that pepo is it's much more common to see than the others. And that one, yeah, they're from all over, sort of the American Southwest as well as in Mexico and Central America. And then the last ones are kind of 2 wild species that we really don't do too much. They're smaller fruit. The Ficafolia, you can see some people will grow that. Okay, pumpkin is grown worldwide. I know here we kind of think of pumpkin pie, but in a lot of other countries, pumpkin is a really important source of nutrients. So pumpkin has, carotenoids. It has a lot of other nutrients that can serve as a, sort of valuable vegetable commodity. And so we're actually number 5 on world production. And if you look in the 20 years between 94 and 2014, production more than doubled across the world. So it's really important. I mean, it's important for our state, but it's important for the world. And our state, we're the pumpkin state. Hopefully you've heard of that. So Illinois produces about half of the U.S. pumpkins, but we produce between 80 and 90% of the canning pumpkins here. And it's hard to see on the map, but basically there's some really, really red spots around Peoria, which is where Morton, Illinois. So Morton is where the Libby's pumpkin processing plant is. And so within a 90-mile radius is basically where the majority of the canning pumpkin in the U.S. is grown. So when you have really bad weather years, that's really problematic for pumpkin production. So for those of you that lived in the state in 2015, you'll know that there was record-breaking rain in late May and early June. More than, this one is saying, 12 to 16 inches. I heard in about a 2-month span it was 2 feet of rain. It's crazy. And so this precipitation at the wrong time, pumpkin doesn't like it so wet. So you could imagine not only is it sort of suffering abiotic stress from having too much water, but that is prime conditions for pathogens. So it was a very, very bad pumpkin year for the state of Illinois. And so of course we're trying to figure out how we can safeguard a little bit more for production. Obviously we can't change the rain, but if there's ways of trying to minimize losses to disease, then that, that's where we want to go with this. And this is actually what the symptoms look like. So of course you have it on the leaves, but the problematic part is on the fruit. It gets this sort of deep pitting into the fruit, and you can imagine since pumpkin is on the field for a long time, you get the bigger problem is the secondary infections that come in and they rot the fruit. And so farmers can't sell this, right? So pretty much any symptoms on the fruit is going to be a problem for farmers. And so this is a pathogen that Dr. Mohammed Babadous here at the university has been working on for, he's been, I think, 18 years, 19 years. He's been working on that the whole time. Over that time, the occurrence and the severity of the disease outbreaks has increased. It's becoming quite a problem in the fields. Probably there is a little bit with management in that some of these farmers don't really do rotations, right? It's not a corn soy kind of thing. And so because this pathogen, it's in the debris, if you plow under the debris, it's going to be a reservoir in the soil, and it can be there for up to maybe 3 years. And so it's quite problematic to get repeat infections year upon year. And of course, every year the titer gets higher. The other problem is it doesn't seem to be managed well with sort of those bactericides like copper-based sprays that normally would be helpful against disease. So it's been a huge management issue here in the state. And so when I came, I thought, okay, there's not a lot of tomato here, which is what I worked on before, so let's tackle this problem. And for me, I'm a molecular biologist, a geneticist by training. We need a genome. There's no genome. Okay, we're going to do a genome. So that's kind of one of the things that we started working on. We got it off the ground in May when my postdoc arrived. And so we were working with the facility here. So we did nanopore sequencing, which is long read technology to get a genome sequence. And then we used Illumina short reads to be able to do error correction. And we got a nice circular single chromosome and 1 plasmid. And so this is just looking at some alignments to one of the closest related species. Our GC content is over 60%, which is pretty common for Xanthomonas. And we had a pretty good assembly. I don't know if there's any genomics people here, but we had a very good assembly. We were able to plot a lot of genes. We did a couple of different ways of doing the assembly, and we got very similar statistics across the board. So we know the genome is about 4.6 megabases. And again, in this particular isolate, which was the type strain, we had 1 small plasmid about 15 kb. So right now, since we got this back, we've been eagerly diving into this genome. There's a couple interesting things I won't go into, but we're starting to work on. So we have a tallow vector, hooray. So we're going to start working on that tallow vector. What is it targeting in pumpkin? It has a lot of repeats, so it's going to be kind of specific. So the more repeats, the more specific it's going to recognize the DNA, because it's a longer DNA piece it's going to recognize, right? So we think it's going to be highly specific, not a lot of targets. So that's one thing we're working on. Another thing that we found, so I showed you the pitting, right? And that pitting phenotype is not really commonly seen in those mesophyll pathogens. So we thought, well, maybe there's some interesting enzymes. And yeah, we found an interesting enzyme. So there's an enzyme that breaks down cellulose. It's an exoglucanase or a biocellulase, I always forget the name, biocellulase, anyway, it breaks down cellulose. I always forget the names. So it breaks down cellulose, and it normally is only present in those that are found in xylem. So you need to be breaking down, those cell walls of the xylem. So the xylem pathogens have that enzyme, but the mesophyll pathogens don't, but ours does. So we're really curious about that enzyme. Is it important for virulence or disease in the leaves, or is it really only important for disease in the fruits? What's going to happen? So we're making mutants, we're doing some studies, and the last thing we're kind of setting up, which hopefully will tides over for a few years is we're actually trying to generate a mutagenesis library. So using a transposon that'll randomly integrate into the genome, we're going to try to collect at least 10,000 individual colonies. Hopefully that will be pretty good coverage across our genome, and then go and screen them, looking for phenotypes, which ones cause less disease. We also have a few others. We're interested in some other signaling, such as quorum signaling or HARP induction, for those who know that stuff. So we have some other phenotypes we're going to screen these mutants for. So hopefully that'll keep my lab busy for a number of years between these projects. But I'm happy about the progress we've made. Again, it was kind of a brand new project coming off the ground, so I think we've made good progress in trying to be able to tackle this disease from sort of a genetic genomic perspective. And my goal is eventually that we can find some effectors in this pathogen that we might be able to target with other resistance genes. So similar to banana, right, where they were using resistance genes from other species, trying to see if they would be effective. That's where we're going to hope to maybe go. And with that, I'm just going to kind of overview. Again, we talked a lot about plant disease and immunity. Hopefully you're going to feel comfortable with that. Genome modifications, there's a lot out there. It's really cool. If you're excited by this, go read. I mean, a new paper comes out, it seems like every week. And then just telling you about sort of the economic importance of Xanthomonas and how it affects such a broad host range, and then what we're doing in my lab on bacterial spot on cucurbits. Anyway, I'm really thankful for my lab that I've been able to pull together. So I have 2 graduate students that are totally mine. So Maria is my PhD student, and then Julius just came a couple weeks ago. And then I also have 2 students that I'm co-advising with Dr. Babadous. So that's Salisu and Sabine, a master's and a PhD student. And it's been great working with them because they do the field work, and so I've been able to learn a bit without doing it myself, because I don't really know what I'm doing. So it's been good working with them, and I had a great group of undergrads that have been working on our projects, and then Ricky, my postdoc, who just came at the end of the spring. And I'm excited because we have a freshman coming under the Ignite program, so I just met her this week. I'm excited to get her on board, and so we're going to be busy. So I thank you for listening to me, for paying attention, and I'd be really glad to go ahead and answer any questions that I can.
What kind of transformation capabilities are available in pumpkin?
That's one of the things we're doing. So there were a couple protocols, more for cucumber. That's a little more common in cucumber. There were 1 or 2 for pumpkin, and we tried them, didn't work at all. So we basically started from scratch, bought a bunch of auxins and cytokinins. We've been playing with ratios. We think we finally found one that's working fairly well. It was the shooting that was the issue. Rooting was no problem. So we finally are getting much more efficient shooting and then rooting. So now, we have some almost to the point to transfer to soil and see if they survive. We had also tried the agrobacterium transformation, and that was no problem. So we did some quick GUS line. We didn't even go full out to the plants, but we could see we could transform in a GUS construct, and they were very blue. So I think we have the parts. We're just finishing up. But yeah, exactly. That's can be the hardest step. So actually, Corey, I didn't mention. So Corey was a senior last year, and he is taking a gap year before he goes to his next adventure. So he's stayed on, and he's doing all of that work for me. A lot of time under the hood.
Have there been any transgenics that have been considered commercializing, deregulating in the cucurbit family?
So there is some transgenics, yes. So there are some lines that have virus resistance, because a lot of places viral diseases are a much bigger issue. So there have been some, I'm not sure if they were, I think they were commercialized. I don't think that they're widely grown, but there were some that were released that had transgenes that were helping protect against viruses. Yes. But again, I was trying to look and see how common they were. It didn't seem that they were really commonly grown. They're not really so much for the winter squashes. They were more for kind of your summer squashes. Yes. As far as, I don't know as much about the cucumber and the melon. I think there are some there as well. Probably more than there are in pumpkin and gourds. There are some. Viruses tend to be the big one that people focus on. They're a much bigger problem. I can look some of those up if you want to get back in touch with me. Yeah. I thought a couple.
Do you think that phylospheric bacterial pathogens affect the distribution and diversity of microbial community, that is endophytes?
Absolutely. And so basically he's asking, when we have, especially I guess these leaf bacteria, right, the ones associated with the leaves, how are they affecting the microbial, so the beneficial or I guess neutral microbial communities? So the mesophyll xanthomonas are going to penetrate through stomata or wound sites in the leaves or the fruit. Now, they're only going to do this when they come to a high enough concentration. So this is part of the quorum sensing. So when they get to a certain density, they'll then turn on their virulence and penetrate. And so you can imagine as they get more and more densely concentrated on the surface of the leaf, they're going to exclude other microbial communities. But at the same time, there are potential biocontrol microbes that we might be able to use to help kind of suppress the growth of xanthomonas. So it can never get to that titer that it's going to be then able, to really penetrate and effectively cause disease. It's kind of both ways, but yes.
And do you think that microbial volatiles are also involved in this interaction?
I'm going to answer yes, because that's probably the answer. I don't know all the specifics, but I do know you get a lot of volatile signaling, even from the roots, right? The root microbial communities, you'll get volatile release, and that will impact leaf pathogens and other leaf communities. It's one of those fields that I try to catch up on. There's a lot coming out nowadays. A lot of people are starting to work on this sort of cross-kingdom and spatially separated communication, right? And so, yes, I'm sure there is. I don't know all the details about it, but yeah, there's a lot of volatile signaling going on.
I did my grad program now several decades ago in xanthomonas, so thank you very much for a really beautiful talk summarizing xanthomonas genetics and everything else you've done. I had the question because my background was rice, and there was, you mentioned the difference between horsicola and horizon, and I wondered, since that's just a few gene differences, is there something in pumpkin that's equivalent, like between that rind, that those spots on the rind versus the spots on the leaf? It seemed to me that I'd never seen a spot on a fruit, on a rind that says xanthomonas.
Right. So it should be the same because, so for instance, I'm looking at Salis in the back. They do spraying of the fields, and so you'll get the same isolate will be causing disease on both. What we think is really going on though with the fruit, it will only infect at very specific developmental stages, and this is actually true with tomato as well. So as the fruit is expanding, you are having cell wall breakdown as those cells expand. And so you can imagine that's going to allow potential penetration areas to get in. Once it's mature, it's not expanding, maybe it's just ripening, it's not going to be able to easily penetrate. And so we think it's probably a pretty narrow window developmentally in the fruit where you can actually get the infection to occur. This is actually a project we've been talking about. We're going to have to do it in a greenhouse and monitor kind of stages post-pollination. But we want to get a fruit assay up and going because that would be really ideal to better understand the infection of the fruit. But it's a big open question. But again, I think that there's probably, the reason we don't see, say, every pumpkin infected potentially is that it can only infect at a very certain stage. So let's say you have a lot of water that week, you get good spread, but then it's dry for 3 weeks. You might not get any real spread of your inoculum during those 3 weeks. So only the ones that were at the right stage during that 1 week where you had a lot of spread, you might get the disease.
Rice, there was the issue of mutation at the end of the silence that was broke out at the end of the leaves that you entered there.
Yes.
And there was also the issue of flax. Those two things, words would be very different.
Well, and again, that's why I hypothesize we would see some different enzymes in the cucurbit ones in order to be able to penetrate through that thick rind. But again, we really need to try to work it out, but we really think once the fruit has reached sort of size maturity, the bacteria still isn't able to penetrate. we'll see. That's something we're definitely going to be looking into. And again, I'm kind of interested in, we don't think that that particular cellulose degrading enzyme should be important for disease in the leaf. So the question is do we see any differences when we mutate it? Like if we knock out that enzyme, does it impact the virulence, the disease on the leaf at all? Or is it really maybe more specific to the fruit? And we can look at gene expression, all that kind of fun stuff.
We have one last question.
To what depth did you sequence for your sample in this assembly, and what assembler did you use?
So yeah, I don't have the depth numbers. I mean, we did do I know it was maybe about, I think, 60x on the nanopore. Am I right on that? I'm looking at my postdoc. Was that about right? We had pretty deep reads in the end. They were quite happy. We tried 3 different ones. So again, I was working with HPC Bio to do this, and they were kind of teaching us so we can do it by ourselves next time. We tried 3 assemblies. So CANU starts with the long reads, assembles the long reads first, and then it error corrects. It seems like it doesn't error correct super efficiently, so we're not really probably going to use that assembly. Spades is one that's really meant for doing these hybrid assemblies, meaning you have long and short reads. So Spades takes the short reads and puts them together, and then it uses the long reads to kind of scaffold that. So you have less errors because the short reads are much less error prone. The other thing that we had noticed in Spades, if you look, we look down here at the number of contigs. So in CANU, there's only one. There's only the circular plasmid. The circular chromosome. But if you look in Spades, we have 2, and that's because it picked out that 15 kb plasmid, and it didn't pick that out in CANU. We don't really know why. We couldn't figure out why. So we're thinking we're probably going to go with the Spades simply. It also had the most genes, which probably was because of that plasmid. There's nothing interesting on the plasmid. So there wasn't much we could really do. Again, it's not very big. My hope is that as we start to do some more looking at population strains, which we're going to be doing as well, so sort of population genomics, we might be able to pick up other strains that have more plasmids. It could be interesting, but at least we had one. Like, okay. But yeah, Spades, I think, is what we're going to end up using in the end for publishing. Awesome, and I'd be glad to answer more questions if you have them during the poster session that's coming up next. Am I announcing it too early? Okay.
Thank you. Okay, so I'd like to invite everybody upstairs for next hour. We're going to have a poster contest. In addition to the judge's choice, we'll have a people's choice. So we want to include everyone here